Hello and welcome to Tomorrow. My name is Lisa and this is our first show of 2019. We've got Dr. Rose Jones from the Bigelow Laboratory for Ocean Sciences, and we're talking about the crazy places where microbes can live. They're living underneath the sea floor and in contaminated mine waste waters. And then Jade, what else do we have?
And then we're going to take a really quick break, but when we come back, we are going to talk about comments from you, the community. So we're going to get into some pretty exciting stuff for the first episode of Science on 2019. Welcome to Tomorrow.Science Discovery 2.01.
And thanks for sticking with us. So like I said, we've got Dr. Rose Jones, an environmental microbiologist from the Bigelow Laboratory for Ocean Sciences. Rose has studied using bacteria to help remediate environmental damage caused by mining. She's also used laboratory-based techniques to supplement natural spring water supplies and has studied microbe communities that live deep within the ocean more than 2 miles below the surface. And also, wrapping it all together, she's kind of looked at how these techniques may help us study extremophiles, or microbes that live in extreme environments, on places like Mars, Europa, Enceladus, and out into the solar system. So Rose, I just wanted to say thank you so much for taking the time to come and chat to us today.
Thank you.
To start us off, I kind of wanted to jump straight into one of the big research projects you've been working on previously, where we take, mining sites and the damage that they cause to the environment and use bacteria to kind of make that better and remediate the environment. But I want to start off by asking, what are these environmental problems that get caused by mining, and why are we actually trying to fix them?
Well, when you dig the rocks out of the ground, you end up leaving some of it behind. So the small amounts of rock ore that are left have different metals in them, and the microbes can eat those metals. There are a particular kind of microbes called chemolithotrophs, and you'll hear me talk about these quite a lot. Those are microbes that literally will eat the rocks.
Oh my goodness.
So they get all their chemical energy from the rocks?
Yes. Wow. And they eat the rocks, and by eating the rocks, they change the chemistry of the environment, which causes the mine waste. So it can be very acid. It's basically like battery acid. It's a big soup of nastiness, which obviously isn't very good for the environment, which is why we then have to then clean it up, because otherwise it gets into rivers, it gets into the soil around, and it starts to poison everything.
So we've got this acid kind of waste coming out into the environment. Is there, say you're at a copper mine, are those heavy metals also being leached out into the environment as well?
Yes, and you can see in the picture you just showed, there's no plants, and that's all because the rocks and the soil has been poisoned so much that there's very few plants that can actually survive there.
Oh my goodness. So it's basically, it's sterilized the surrounding environment?
Yes.
And
it's poisoned everything.
Oh my gosh. And is there any hope to regenerate the ability to regrow anything there?
There are certain ways that you can do that, and that's one of the reasons that I've been looking at the remediation. But that's what that means. Remediation means cleaning up the environment. And you can do it, sometimes you can use certain chemicals, but the way I've been doing it is using bacteria, because you can do the same job without throwing a load of chemicals at the same thing.
Got it.
So what kind of bacteria did you introduce into this mining waste environment, and why did you choose that particular species of bacteria? I'm guessing you can't just use any bacteria that's out there. You've got to pick one that is going to work for that environment.
Yeah, the way this particular microbe actually came from the environment that we wanted to clean up. an ancient copper mine in about an hour away from my hometown called Paris Mountain, or Mynydd Paris. And it was once the largest copper mine in the world. So it's been poisoned for 4,000 years. And in that time, the microbes, there were microbes that had adapted to living in these really harsh conditions. Mm-hmm. And we managed to isolate one of those that would grow as long streamers. So it looks like long strings of slime. And we took it out of the environment and then stuck it. If we change the environmental conditions that we give it in the laboratory, we can get it to, instead of working against us, to get it to work for us. And instead of putting the metals into the water, we can get it to take the metals out of the water. Mm-hmm. So the beauty of this one was that it was a native to the environment, so we weren't even introducing anything.
Nice, and that was actually going to be my next question. So it's already so it's already natural to the environment, but is there any potential for like an overgrowth? Say these bacteria get a little too comfortable, and is there any potential like negative effects that a lot of these bacteria can have?
I mean, that's the whole point of the acid mine drainage is caused by these microbes getting too comfortable. So the problem's already there.
Got it.
Where we did it was take these microbes out and put them in we would put them in a separate tank. In this case, in the laboratory. And so they were contained, and we could then change the conditions to get them to do what we want.
Got it.
A lot of these systems, you will put them in a tank to keep them separate.
So I've got another question here. This one comes up from our chat room, actually. It comes from Astro YYZ, who's watching on Twitch. And they want to know, can bacteria be used to clean up other polluted areas in things like oil leaks?
Yes. You can do the same thing. So there's some research going on, which isn't my area of expertise, but there's research looking at doing the same thing for oil microbes. So taking a microbe that we know eats the oil, making lots of it, and then feeding it the waste oil. So then it will eat the waste oil for us. There's also some research looking at recycling e-waste, so electronic waste, in the same way, getting the microbes to dissolve and eat and change the chemistry of the waste, so to peel off things like copper from wires and gold from circuit boards.
It sounds like there's a lot of different impactful ways that bacteria can help us clean up our planet and make it a little better for all of us. But with your research in the acid mine drainage and that bacterial system that you said was working in the laboratory, is there a plan to like scale that up and like take those tanks that you had in the lab and move them out into the field and kind of get that going like in situ?
That particular one, my particular work, not at the moment, but there are similar sites popping up all over the place. There's one in a mine down in Cornwall that was running for a while, and you can use the same sort of technology in all sorts of different places. You can also use it to extract metals from the rocks in the first place. So you can use them to mine the metals and then clean up the waste afterwards. And there's a lot more of that going on. I think it's something like 20% of the world's copper is made by using microbes as miners.
Mm-hmm. So, and all of this, I mean, it's a very, I mean, at the end of the day, like what you're doing is extremely important, and it's, I mean, especially with the, very, very rapidly degradation of our environment. So what kind of led you down this path to discover these or to work on these really clever solutions for things like pollution and whatnot?
I like microbes that live in weird places, and the weirder the better. But I also, in working with these microbes, I could see that there are certain questions that we can use them in order to, they can provide a sort of an answer. It's a lot more work on the front end, so you have to do an awful lot more thinking and designing rather than throwing just a chemical in. But when what you get out of it is a system that's usually more, it takes less energy to run, so it's more it's more sustainable, I should say.
Coming back to microbes that live in weird places, because that's what you like to research, I want to flip for a second here over to microbes that live kilometers below the surface of the ocean and actually within the rocks that make up, the oceanic crust. Because here I am thinking, okay, when I first read this, I was a little bit confused because I was like, hang on, all right, we've got the sea floor. I've seen images before of like black smokers where, hydrothermal vents where the volcanic processes are like spewing up gas and stuff from the bottom of the crust. But then I heard that there are microbes living within the rock of the crust. Like, isn't that solid rock? Is this actually possible? Or are there actually microbes living that far underground?
Yes, we know there are cells and we can see them. But when you think about the actual crust of the Earth, it's not actually just a single solid block. It's actually full of cracks. And there's water flowing through those cracks. Just like you would get water in your groundwater in your basement, it's exactly the same in the Earth's crust. And if you think about the hydrothermal vents, the black smokers, there's water coming out, so it must have gone in somewhere.
Oh, yeah.
And this water then percolates through the Earth, and it changes as it goes along in the crust. And that's how it ends up spewing out with all the metals, which is actually what the black is not just smoke, it's the metals falling out of the very hot water as soon as it hits the cold seawater. And that water's got to have come from somewhere. So, and the microbes then, the cells attach themselves to the rock and they have the water flowing through. So it's not just a solid block.
So we have water coming down through these cracks, and we know that there are microbes living on the rock there because of research groups like yours that take trips out to the middle of the ocean and actually dig down and find them. So for your research group, you actually took a trip out to the Mid-Atlantic ridge. Could you tell us a little bit about that trip?
Yeah, it was 4 weeks at sea. We had to load everything we needed onto the ship, because when you're in the middle of the ocean, you can't really just go to the shop and get whatever it is that you forgot to pack. And you then spend days and days steaming out to the middle of the ocean. And when you get there, you've then got to get through 2 miles of the deep sea. And to do that, you'd need a submarine, or in our case, it was a remote operated vehicle. So those are robots which you take, and then you have like a cord that attaches to the ship, and the pilot's there telling the robot where to go. And they have like these joysticks. They're awesome. They can get these robots to do everything, and then they have the arms and they manipulate what you want them to do. And at that point, we were going back to an experiment that had been installed 6 years previously. So we went back, and our samples were suspended in holes in the seafloor that had been drilled. So we were there to go back and pull these samples out, just like a cork from a bottle. And then these samples were small pieces of rock, which we'd left there as a sort of like cheese in a mousetrap, essentially, so that the microbes would come and collect on the rocks, and we could then pull them out of the seafloor and take them with us. Because trying to get a single cell that you're trying to study from the bottom of the deep sea all the way up to where you want to study them can be quite a challenge.
What was that feeling like when you guys, you take 4 weeks at sea, you finally reach your study site, you get, because it's a whole process, right? You get the little undersea robot all the way down, you get to the sample well, you finally be able to bring those samples back up to the surface. And what's the feeling there? Is it a giant sigh of relief, or is it, oh no, now the samples are in the air, we need to like get them quickly processed so they don't degrade or degrade over time? Like, what was the process once the samples reached the surface?
Well, it's definitely more the second than the first. It's like, yay! First of all, you've got to, because we're dealing with things which, it's like, do you get your samples up at all? A lot of the time when we were taking up these samples, sometimes it would break off, parts would break off, so we'd only get half. Or in one case, we pulled it up and the weights broke halfway, so we lost it and then spent a few days trawling the seafloor trying to find it. And he was like, do we find these samples? And we found them. It was awesome. It was really exciting when it came up. We were like, yes, we've got everything back. And then, of course, you've got to hurry to make sure that these things aren't exposed to the air or just to us, because there are microbes on us. So you have to keep them as cold and as wet and as sterile as you possibly can. So everybody's running all over the deck. There's bits piled everywhere. We've got tools trying to pull everything apart. And these things have been sat there for 6 years on the seafloor, so they're full of gunk and you get covered in muck. And it's, it's a bit frantic for a while. You definitely need hard hat and a boiler suit because you get covered in slime. But then after, and at this point, that particular site, they came up at like 3 a.m. in the morning, so you're half asleep. But by the time it gets to lunchtime, everything has calmed down, everything's in the fridge, you're like, finally. And then you go and have a nap and then do some more samples until the next day when you have to get up at 3 a.m. to do everything again. So it's quite manic.
Absolutely.
It's great fun.
Yeah, so pretty involved process obtaining the samples, but what happened to them once they made it back to the lab?
I've been looking at them using a particular technique called bioelectrochemistry.
Oh, wow.
And what that means is these microbes like growing on the rocks, but because they like growing on the rocks, they're very hard to study because it's hard to separate them from the rock. The only really way you can tell if they're doing anything is by counting the cells. But if you're counting a cell, you don't really know whether it's alive or not because it's just sitting there. So what I've been doing is we know there are certain kinds of microbes that can they can eat the rocks whilst they're still solid. So they might be eating iron or sulfur or whatever, but that's inside the rocks. So the energy is coming directly from these solids via just electrons, basically. And all chemistry and all getting of energy and all of eating comes down to moving electrons around. And we can also give them electrons via electricity. So I've basically been plugging my machine into the mains power, changing the energy level so that the microbes think that they are eating iron when they're actually eating just electrons. And from that, we can measure changes in the electrical output. And from that, we can tell whether the microbes are growing or not. So it's a way to track the growth of these microbes, which would otherwise be very difficult to study.
Are there different groups of microbes that prefer different voltages? Like you mentioned, you can trick them into think they're eating iron, but are there some microbes that prefer eating sulfur compounds instead of iron-based compounds?
There are certain microbes that, yes, will eat sulfur or iron or whatever else, but it's kind of a little early to tell whether we can do that via bioelectrochemistry, because to start with, no one's ever really done that in the deep sea the way I'm trying to do it, so we can't really say for sure. But when we look at similar systems we know of on land, it might be possible, but again, we haven't really done enough research to be able to say that because it's still in the very early stages of figuring out how this works.
Absolutely. So we actually have a pretty interesting question from Twitch on our chat from Alien Mind. And they're asking, can you teach those microbes to survive better on Earth's surface or other environments?
Usually, we don't teach the microbes that we're finding particularly, because if we want to get a microbe from a particular environment, we will go and get it from that particular environment. Because microbes are everywhere.
Yeah, exactly.
So it's quicker and easier at this point to go to the environment that's similar to what you want. So for an acid mine drainage system, you would go to another acid mine drainage system. But as technology moves on, we might be able to design and transfer characteristics to make that happen.
Yeah.
But at this point, it's still easier to go and get the microbe entire.
Yeah, and when you think about it, that's kind of like pressing the fast forward button on the whole evolution thing, because I mean, these microbes have evolved to adapt to their environments. But if we are, as an external source, are able to just manipulate them or, you know, kind of make them adapt pretty quickly, then I think, yeah, the repercussions of that would be pretty profound.
Exactly.
Yeah.
I want to talk a little bit more about, you mentioned, you know, the work that you're doing hasn't really been done. These microbes haven't really been studied in depth that much before, which is why you guys are going to these extreme environments and trying to find them. Why do you think those microbes are in that particular ecological area? And what do you think would happen if, you know, if we made a habit of deep sea mining, for example, mining the sea floor? Now we know that stuff's, you know, actually down there growing. Are there any potential repercussions on a larger scale that could happen if we keep disturbing these microbes that we don't really know that much about?
Yeah, that's a really good question, actually, because we don't know what would happen. We don't know if these microbes, because these environments are very cold and dark and there's not much for them to eat, they may not be doing very much. So it might be that they never do very much. But on the other hand, it might be that they do a lot just because of the amount of seafloor that there is that they could be working in. So if we disturb them, we might be disturbing all sorts of cycles like burying of carbon or releasing of nutrients into the sea. And there's also, with the deep sea mining in particular, we don't know if we end up with acid mine drainage on the seabed. We don't even know whether that will be a thing or whether the seawater will be enough to dilute it. We don't know any of these things. But if we go there and we interfere with the environment before we understand it, then we risk creating a whole load of problems for ourselves that we don't know how to answer yet. And we also risk just destroying things before we even knew that they were there.
And that happens to be a point of contention that comes up on this show a lot, actually, is when we're talking about you know, all of the big plans we have for Mars. But as you said, like, even though life might not exist on the surface, it can definitely, there's a possibility it can be below surface. And so that's kind of like a debate we have ongoing between the hosts on this show is, well, you know, it's like you said, we don't know what detrimental effects we could potentially have. I mean, we could potentially wipe out like a little tiny ecosystem for all we know. So that must be really interesting, having to kind of balance the uncertainty with the benefits of the actual research.
Yeah, I mean, for us, it's slightly easier in that we know we can see the cells, we know what the cells look like. If you're talking about Mars, you might not even know what you're looking for.
Exactly.
I think that's a great segue into the last part of this interview that I want to get to is these microbes that are living so far down beneath the sea. I'm guessing they're exposed to, you know, really high pressures and temperatures that are very different than microbes that we usually think of. And so these are really extreme environments. And is what we're learning from studying these microbes going to help us study microbes on Mars that we're looking for, or on Enceladus or Europa? You know, how do those two things relate to each other?
there's an awful lot of parallels between studying the microbes in the deep sea and how we think about studying life on other planets. To start with, we think about deep sea as being, like you say, cold and dark and really extreme, and also really remote and full of weird and wonderful things. And we think about it in a very similar way as we think about what we might find on other planets. And because these are so extreme, the idea is that the laws of physics and chemistry are going to apply the same on Earth as to anywhere else. So, at least as we understand them at the moment. So, if we can find life living at very low pressure and very high temperature and what have you on Earth, then it's a good chance that we could find them elsewhere. If we can't find them, then maybe it's less likely that we would find them there. And because space is so very big, you can't just, it's like looking for a needle in a haystack. So the more we can target where we're looking for and be intelligent about where we pick these things, the easier it would be to concentrate our resources on finding where it's most likely to be.
Well, hopefully we can take all those lessons learned and hopefully end up finding life on other places sometime. We got a lot of questions coming in from the chat room here. They're actually not, they're related, but everyone is really excited about bioengineering, which is a little bit kind of different than what we've been talking about, but I'm just going to dive right into St. Alex. Coming back to you feeding your bacteria electricity, St. Alex wants to know, if microbes like electrons, do you think that they could be used as growable fuel cell membranes?
Yes, there's actually quite a bit of, in fact, most of the research on bioelectrochemistry is looking at fuel cells. And you can find these microbes in mud and everywhere. So there's an awful lot of research being done on, okay, well, if we have a wastewater plant with a load of organic waste, can we tweak the conditions to get these microbes to grow? So whilst they're eating our waste, they're also kicking out electrons, which we can then capture and convert into electricity. Each microbe won't generate very much, but if you can get enough of a density of them, then you can start to generate measurable amounts. It won't solve all the problems, but it can help.
Interesting. And then going back to the application of this, Slapstick Mojo on Twitch asks, although microbes and sickness are often a problem in space, could there be any major benefit in spacecraft system use?
See, now there you're shifting into things which I don't know. I wouldn't say I have a whole lot of expertise in.
Okay.
But one thing I could say, is that it depends on the problem that you're asking. Mm-hmm. If you're talking about getting metals from a mine, say you're thinking about colonies on wherever you happen to be, and you want to get these metals out of the place that you're living rather than shipping them from Earth, then I could say, well, yeah, maybe you could, because there are microbes that grow in acid mine drainage conditions that don't need oxygen. So you could set them up and they might be quite happy doing that. Mm-hmm.
That'd be
but
really
that's
neat
way in the future.
Absolutely.
Yeah. many years of research down the line, but I mean, it could be a really nice way to take microbes and bioengineer them into use cases where they benefit us, maybe even on Mars, to help us mine metals and ores. And coming back around to bioengineering, I want to bring up, it's more, I guess, a comment rather than a question, but I think they raise a really good point, and I want to get your perspective on the, I don't want to say ethics, but like the what we could be getting ourselves into by bioengineering. So Elmar says, I'm not sure if this has been mentioned, but while this sounds quite interesting and useful, I'm reminded of the novel Wind by Kevin Anderson, and And basically, an engineered microbe that consumes oil is released to clean up a massive spill. But it ends up taking out all oil and plastics. Far-fetched, yes, but could it happen? So do you think we're at a point where we understand enough about microbes and their ecology and the way they function to be able to be messing around with these ecosystems to kind of benefit humans? Or do you think we have a little bit more to learn before we should start engineering these microbes or putting them in places where they don't naturally live?
That's a really good question. And it's definitely worth thinking about these things. When we're thinking about putting, for example, the acid mine drainage systems, one of the things that we will do is try and keep them, at least in the systems that I was dealing with, we were thinking about that in terms of taking the native microbe out and putting it in, or keeping them in tanks and things. But it also depends on the environment that you're talking about. So if you think you understand enough about the environment, then maybe you could make that judgment. But there's an awful lot of environments where we just don't know enough to be able to say one way or the other. But it's certainly something that's worth considering when you're weighing up whether a project's worth doing or not, or at least whether we should do it or not. Although I will say that humans are already changing the environment so much that in some ways it's a case of, well, how do we get the environment back to how it was before we messed it up, like with these acid mine drainage environments, with these poisoned landscapes. And at that point, it's, well, we might be introducing something else, but it will also hopefully make it better. So does that balance it out? It's a really interesting question, though.
So you've already done so much fascinating research. What are some of your future plans for research or for this project?
The next steps, at least for my research, I'm continuing to look at the microbes in weird places, and particularly the microbes that eat the rocks, the chemolithotrophic microbes, because I think they're very interesting in there are so many potential things that you can do with them, but also there's an awful lot of rock out in the environment. So, and they grow in all of these weird places. So if we can say, okay, these microbes grow in this weird place, can they grow in this really, really weird place? And there's just so many weird places that they could be growing. And I'm like, I want to visit them all and find out.
Fantastic. And if people wanted to find out more information about you, Rose, and also the Bigelow Laboratory for Ocean Sciences, where can they go?
There is the Bigelow website if you want to find out more about the laboratory where I work. And I also have a website and a Twitter account, which I believe you have the contact details for. So feel free to contact me on that if you want to know more.
Wonderful. Once again, thank you so much for taking time out of your Saturday to join us and talk to us about the incredibly strange places where microbes live. It's been an absolute pleasure, and thank you so much again.
Thank you.
All right, what we're going to do is, before we take a quick break, we want to thank the people that actually make these shows possible, specifically our Escape Velocity Citizens of Tomorrow. These people contribute $10 per episode to help make these shows go, to help bring us amazing guests like Rose, and to help us share incredible stories of science and how science actually gets done. We also have our Orbital Citizens as well. These are the people that contribute $5 or more per episode, and they help make these shows happen, and they also get a host of amazing awards as well. So if you'd like to know what those rewards are, head on over to patreon.com/TMRO. Now we're going to go ahead and take a very quick break. Please stick with us, because when we come back, we're featuring comments from you, the community of tomorrow. See you soon.
We've always looked to the stars. They guide us, give us comfort, help us find our way. We see ourselves out there. When we look up, it inspires us. And we long for something we don't yet know. We yearn to go there. So, we venture forth.
We choose to go to the moon in this decade and do the other things, not because they are easy, but because they are hard. Because that goal will serve to organize and measure the best of our energies and skills.
Tranquility Base here. The Eagle has landed. It's one small step for man, one giant leap for mankind.
The exploration of space will go ahead, whether we join in it or not.
Many think we stopped exploring. But we know our journey didn't end.
T-minus 1 minute.
We've only just begun.
Orion is functioning perfectly at this point.
Come with us and explore tomorrow.
Hello, and thanks for sticking with us. So we are going to go ahead and dive right into your comments from our previous episode, which is where we got to interview Dr. Kirby Runyon from the Applied Physics Lab, and we got to talk all about Pluto, and that was such an amazing interview. You guys loved it. We certainly loved it.
I learned so much about Pluto.
Oh my God,
Like,
I got so excited.
There was like ice spikes that like have fallen over to a particular angle.
Gosh. Yeah, exactly. And I'm pretty sure TMRO made a lifelong friend of Dr. Runyon, so that was always lovely. But let's just go ahead and dive right into this comment. It's a little bit long, but bear with us. It's from YouTube, from Crane Army, and they state, "For someone that never really cared about whether Pluto was a planet, the way its status was changed never really felt right to me either. This guest gave me the language and the reasoning to explain why it didn't feel right after many years of simply putting it out of my head. Dr. Runyon also made the point about how language has the ability to change perception. The history of the color blue and language is an interesting case study on this. He's given me the language to realize a perspective and articulate it beyond throwing a tantrum in frustration. That is a rare and powerful gift."
So the reason I put this extremely long wall of text into the comment section, and bear with me, I don't usually do walls of text. I usually cut things down. But I felt like this comment really kind of hit home the message of how language is really important in how we see things in science and how we define them. And they mentioned the example of the color blue. And recently I watched a video about how the color blue in different languages of a few First Nation people, they don't have a word for the color blue, but they have about 20 words, for example, for the color green. And so there's this debate going on whether did these communities have the ability to actually see and perceive the color blue, or did they just see it as another shade of green? And there's not enough evidence either way to see whether did they actually have the like receptors, cells in their eyes that could detect that blue wavelength of light, or maybe they could, but they just didn't have a word for it. Because they didn't have a word for it, they just segregate is not the word I'm looking for, categorized it as something that they were already familiar with. And so I think that's important when we look at things like planets in our solar system. Are we going to categorize something as something that we're already familiar with, or do we give it its own category? And how does that affect how we view those objects? I think it's just a really insightful thing to think about, and a perspective I've never considered before. So I don't know if that makes any sense to you. Maybe I'm just ranting about blue and green right now.
It's
but
so interesting. Yeah, no, definitely. Because I mean, that like you said, it all comes back down to perception, and it all comes back down to ultimately, you know, how we view our home, which is the solar system.
Yep.
And I think that Crane Army brings up a really beautiful point about how, especially when it comes to the Pluto debate in particular, there's a lot of like passionate, like at least at one point, there's a lot of fervor. Like people were really offended and like personally hurt by Pluto's demotion. But what I loved about that interview was he really did put context to it, and he put it into perspective. And like you said, or like Crane Army had stated, it gave you the language to be able to put more words to those feelings. And it's so therapeutic and so cathartic in a way. Because yeah, I mean, for a lot of people, while we're not yelling at Dr. Tyson, there's also something, you know, inside of us that just didn't quite sit right. And he articulated it so well. And the great thing about that interview is, yes, we talked about all the amazing science, all of the amazing data about Pluto itself, but we really got into kind of a more philosophical conversation about how important classifying things are and how important it is to be able to understand something thoroughly enough to be able to kind of create like a cohesive system of like, this is what this is what this is, and how that does kind of evolve over time.
I think it was important that we heard that side of the debate from someone who is actually a planetary scientist and works on these things. You know, it really added credibility and scientific merit to that argument of for and against, yes planet, no planet kind of thing. So we've got Launchpad Astronomy, or Christian, who talking about New Horizons and Pluto. Christian did an amazing 24-hour Ultima Thule live stream when New Horizons flew past Ultima Thule. Thank you, Christian. We had so much fun on that stream. Thank you for being in our chat room. But Christian did want to say that words mean things to people. Also, classification helps us think about what sort of questions we can ask. So I completely agree. I think you've hit the nail on the head there. Once you give something a label, a category, that defines what kind of studies you do on it and the curiosity that you have about that object. So. All right. So who do we have on next week, Jade?
So this is a very exciting time of the year because you get science back to back. Week to week.
2 weeks in a row.
February 2nd, which is next Saturday, we're going to have Mary Michael Cherrier.
Yep.
Yay, Mary Michael Cherrier, Scientific Engineering Associate from the Lawrence Berkeley National Laboratory. And we are going to talk all about synthetic biology and the modification of bacterial surface structures. So we should be getting into some pretty fascinating stuff. But before we close out the show officially, I want to give a heartfelt thank you to the folks who make this show possible. Yes, I am talking about the Escape Velocity citizens. These generous folks contribute $10 an episode on Patreon, and we also have our Orbital citizens, $5 an episode on Patreon, and we're going to keep going down. Suborbital citizens, $2.50 per episode on Patreon, and last but certainly not least, our ground support citizens. Yes, you saw that font size get several times smaller. These folks contribute $1 per episode on Patreon, and just go ahead and try to take a look for your name.
Might take a while.
I'm sure you have. Okay, great. So a big thank you to all of the folks previously mentioned, and thank you for tuning in to this episode of Tomorrow's Science. We hope to see you next week, and stay curious.